Most drugs act on a cellular level by chemically binding to receptors on cell membranes or inside cells. The cell membrane contains receptors for many kinds of substances such as hormones and neurotransmitters. Some receptors also interact with drug molecules. As a drug molecule binds to a receptor, reactions are initiated that stimulate or inhibit regular cellular functions. One type of reaction activates, deactivates, or otherwise alters intracellular enzymes. Almost all cellular functions are catalyzed by enzymes. Therefore, drug-induced alterations can greatly increase or decrease the rate of cellular metabolism. Another type of reaction involves changes in the permeability of cell membranes. A drug molecule binds to an ion channel receptor, a receptor protein, that is a structural component of the cell membrane. The drug's binding may open or close the ion channels to allow or prohibit the movement of ions into the cell. In nerve cells, for example, movement of sodium ions into the cell usually excites the cell. Movement of potassium ions out of the nerve cell inhibits nerve cell excitability and function.
Aldosterone is a lipid-soluble hormone that can easily diffuse through the plasma membrane. Inside the cell, aldosterone binds with an aldosterone receptor molecule in the cytoplasm. The aldosterone receptor complex moves into the nucleus and binds to DNA. The binding of the aldosterone receptor complex to DNA stimulates the synthesis of messenger RNA, which codes for specific proteins. The messenger RNA moves from the nucleus into the cytoplasm and binds to ribosomes, where it directs the synthesis of specific proteins. These proteins produce the response of the cell to aldosterone. The sodium channel has receptor sites for a ligand acetylcholine. When the receptor sites are not occupied by acetylcholine, the sodium channel remains closed. When acetylcholine binds to its receptors, the channel opens. Therefore, the channel is called a ligand-gated ion channel. When the channel opens, sodium ions diffuse through and enter the cell. The sodium channel has receptor sites for a ligand acetylcholine. When the receptor sites are not occupied by acetylcholine, the sodium channel remains closed. When acetylcholine binds to its receptors, the channel opens. Therefore, the channel is called a ligand-gated ion channel. When the channel opens, sodium ions diffuse through and enter the cell. Has a receptor site for a small molecule, called a ligand, exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of cAMP from ATP. The cAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.